Okay, there are several types of naturally occurring RNA in prokaryotes and eukaryotes. So we have the most abundant, the ribosomal RNA, which constitutes 80 to 90%, and it constitutes two components, the large and the small component, and these components are detected using agarose gel electrophoresis. We also have uh, the one which constitutes only 2.5 to 5%, the messenger RNA, and it is detected as a faint background underlying the ribosomal RNA and is visualized by agarose gel electrophoresis. We also have the other smaller, very minute amounts of RNAs, transfer RNAs, and small nuclear RNAs and other RNAs. So for the extraction of total RNA, so the preparation of specimen material for RNA extraction is somehow different from DNA extraction because, for example, if the reticulocytes in blood and bone marrow samples, they must be lysed by osmosis or separated from WBCs by centrifugation. And in cases of tissue samples, tissues must be dissociated and kept frozen in liquid nitrogen or immersed in buffer so that these steps will inactivate intracellular RNases. Specifically for the pancreatic tissue, which contains large amounts of innate RNases. And if we're looking for bacteria and fungal RNAs in samples, then isolation by chemical lysis or by grinding in liquid nitrogen. And viral RNA in serum and other cell-free fluids. Usually, the isolation is performed especially in formulated spin columns or beads. So this slide shows the steps of how to extract total RNA. So the steps are as follows. We have cell lysis, protein extraction, phase separation, precipitation of the RNA, pellet formation of the RNA, and the washing and resuspension of RNA. So cell lysis is performed by using detergents or phenol in the presence of high salts in sodium chloride or in RN or with RNase inhibitors. And we can also, instead of using high salt buffers, we can have guanidine thiocyanate because this is a strong denaturant of RNases. We also have 2-mercaptoethanol, which is a strong reducing agent. We can also utilize that. And then we must add DNases and RNases to remove contaminating DNAs and RNAs. So for the protein extraction, here we will be using acid phenol in combination with chloroform and isoamyl alcohol because this combination of solution can efficiently extract RNA. And what is the function of chloroform here during protein extraction? It is used to accentuate or enhance the extraction of the nucleic acid because it can denature proteins and hence it can promote the separation of phases. And to prevent foaming, isoamyl alcohol is added. And then still under protein extraction, for RNA, the organic phase must be acidic, and because the acidity of the organic phase can be adjusted by how can we adjust the acidity? We need to add buffer and the of the appropriate pH. And then for the phase separation, the upper aqueous phase containing the RNA is removed to a clean tube. So always remember that it is always in the upper aqueous phase or layer that we can observe the DNA or RNA, and then not the lower hydrophobic layer. And for RNA precipitation, we can do this by adding 2 volumes of ethanol or 1 volume of isopropanol. And glycogen or yeast transfer RNA can be added as a carrier. See, this is a carrier for pellet formation, the glycogen or yeast transfer RNA. And 70% ethanol is used for RNA washing, and the RNA is a free buffer or water for RNA resuspension. So, a more rapid and usually efficient method of DNA extraction involves the use of solid matrices for binding and washing the DNA. So, we have silica-based products have shown effective DNA binding capabilities in high salt conditions, and various applications or adaptations of this technique has been developed, including the utilization of diatomaceous earth as a source of silica particles. And modern systems are available for purchase featuring solid matrices in the form of columns or beads. So the steps include, before the lysate is applied to the column, this is the column, the strong denaturing buffer conditions must be adjusted. And then it is followed by the addition of ethanol. We also have the filter columns, columns with filters. These are for the removal of particulate materials before application to the adsorption column. And commercial reagents are applied with the columns to optimize RNA absorption and washing on the silica-based matrix. And then lysates are applied to the column in a condition wherein there is this high salt chaotropic buffer. Washing of the adsorbed RNA is with buffers. And then, of course, DNA has to be added. And then the washing solutions and the want are drawn by the column by gravity, vacuum, or centrifugal force. So always remember the small columns of silica-based material that fit inside microfused tubes or spin columns are widely used for routine nucleic acid isolation. So generally, guys, 1 million eukaryotic cells or 10 to the 50 mg of tissue will yield about 10 UG of micrograms of RNA. So here, RNA yield from various sources. So since our RNA yield isn't that much, it is better that we are vigilant in handling the specimen properly. Because C and ml of blood will only give us 1 to 10 micrograms of RNA. And then remember that specimen handling specially affects RNA yield. So the isolation of poly-A RNA will result in a much lower yield, and always RNA yield will depend on the WBC count, the type of cells, and the conditions of the cell culture. And we can also get more RNA from liver, spleen, and heart tissues as compared to brain, lungs, ovary, kidney, or thymus tissues. And the isolation of RNA from fixed tissues is especially affected by the type of fixity. So the isolation of poly-A or messenger RNA. So the limitations to the isolation of poly-A RNA using oligo-DT or DU. So what do you mean by oligonucleotides, and DT for deoxythymine and DU for deoxyuridine. So again, oligonucleotide, deoxythymine, and deoxyuridine. So these are the limitations. So the efficiency of poly-A and poly-U binding is variable. The secondary structure in the target sample may compete with binding to the capture oligomer. Messenger RNAs with short poly-A tails may not bind efficiently at all. And AT-rich DNA fragments might also bind to the column and not only compete with the desired messenger RNA target, but also contaminate the final one. There's also potential digestion of oligo-conjugated matrices, and rRNA can co-purify with the poly-RNA. So it will result to a final purified product. However, enriched in poly-A, but not a pure poly-A preparation. So those are the limitations that can be encountered when isolating poly-A RNA using oligo-DT or DU. So as mentioned earlier, approximately 80 to 90% of the total RNA comprises rRNA or ribosomal RNA. In many cases, the RNA needed for analysis is messenger RNA, constituting only around 2.5 to 5% of the overall RNA yield. And the majority of the RNA originates from highly expressed genes, with rare or single copy mRNA representing a minimal portion of the total RNA population or isolation. So, to enhance the yield of mRNA, particularly for rare transcripts, protocols frequently utilize oligomers of thymine or uracil immobilized on a matrix, resin or column or beads. And then the poly-T or poly-U oligos selectively bind to the poly-A tail found exclusively on mRNA. So the poly-T or poly-U will selectively bind to the poly-A tails. And these poly-A tails are only found on mRNAs. So measurement of nucleic acids, quality and quantity. So we can measure nucleic acids using electrophoresis, spectrophotometry, and fluorescence spectroscopy. However, we should remember that if we're having electrophoresis, that's a qualitative approach. It's not quantitative. And this is the one that we can perform this in our laboratory and spectrophotometry too. And please take note that spectrophotometry and fluorescence spectroscopy are both quantitative, so we can really have values for such here. So electrophoresis involves nucleic acid resolution using agarose gel. So that's why we have the agarose gel electrophoresis. And you might wonder, how can we visualize the nucleic acids? So by using fluorescent dyes, of course. For DNA, we can use ethidium bromide and cyber green 1. For RNA, we can use ethidium bromide still and cyber green 2. And for DNA only, we can also use silver stain. While for spectroscopy, the principle for this one, it uses the Beer-Lambert's law. So it's more on the relationship of the nucleic acid concentration to absorbance. That's why we have absorption constants. 50 for DNA, 40 for RNA. So using the absorptivity as a conversion factor from the optical density to concentration. So meaning one optical density unit or absorbance unit at 260 nanometers is equal to 50 mg per liter or 50 UgML of DNA and 40 Ug per ml of RNA. So that's it. Here, where A is for absorbance, epsilon or E for molar absorptivity. We have the B for the path length and C for concentration. So the absorbance at this wavelength is thus directly proportional to the concentration of the nucleic acids present in the sample. So again, spectrophotometric analysis for the quantity of nucleic acids is based on the relationship of the concentration of the nucleic acid, the concentration of the DNA or RNA to absorbance. And we also have fluorometry. So it measures fluorescence related to DNA concentration in association with DNA-specific fluorescent dyes. So early methods use DABA, and DABA combines with alpha-methylene aldehydes to yield a fluorescent product, binds to the deoxyribose. And the modern methods, we have, I don't know how to read this because this is German, it's Fish 33258. It combines with adenine thymine base pairs in the minor grooves of the DNA double helix, and Fish is specific for intact double-stranded DNAs. So here we have the products, the stained products under electrophoresis. So the first one, the black and the gray, black or white picture depicts the staining of the gel using ethidium bromide. So this agarose gel is stained with ethidium bromide, and these are the bands. Here we have the first column represents the markers, and these are the bands of the isolated DNA or RNA products. We also have the second picture on the first column. We can observe green bands, hence cyber green. And the third one, the yellowish brown-like color, is an agarose gel stained with silver stain. So stained with silver, sorry. And under spectrophotometry, it's not colorful, but it's just here the DNA is quantified with the nanodrop spectrophotometer, and the DNA concentration is the concentration is can be of course detected. And the good thing with the nanodrop, because it can tell us whether there's a potential contamination from RNA and proteins if we're doing DNA quantification. And here we also have, for under the best example, modern example of the, please don't get confused, because under electrophoresis, we're also using fluorescent dyes. Of course, under fluorimetry, we're also using DNA-specific fluorescent dyes. So here the example is the Hirsch. Hirsch dyes are a group of fluorescent stains that are commonly used for DNA binding in biological lab applications, and these dyes are particularly known for their ability to selectively bind to the minor grooves of double-stranded DNA. And the binding of Hirsch dyes to DNA results in fluorescence, making it a valuable tool for various techniques such as fluorescence microscopy and DNA staining in gel electrophoresis and fluorometry. Because when Hirsch dyes bind to DNA, they undergo fluorescence enhancement, emitting blue fluorescence under UV light. And these dyes are widely used in cell biology and molecular biology research to visually, this is visualization, and aside from visualization, there is quantification of DNA content in cells. And additionally, Hirsch dyes are employed in DNA staining protocols for distinguishing between live and dead cells, or identifying nuclear morphology in cellular studies. So under electrophoresis, so the visualization of DNA in agarose gels is influenced by the type of isolated DNA. So a well-prepared plasmid DNA sample, this sample typically exhibits a bright singular band of supercoiled plasmid DNA with moderate mobility, so it will migrate downwards, accompanied by minimal or no additional bands representing nicked or broken plasmid forms. However, genomic DNA with high molecular weight is expected to appear as a distinct band with low mobility near the top of the gel. So this is the top part of the gel, and the mobility is not that much. It haven't migrated that. So meaning, for example, if this is a high molecular weight genomic DNA, it will just stay near the wells. So that's what we call low mobility. For high quality RNA preparations, we have two clear bands of RNA are anticipated. This one and 2. Remember the small and the large component. Two distinct bands of ribosomal RNA. And the integrity of these bands serves as an indicator of the overall RNA integrity within the same sample. So if these bands display degradation or smearing or are absent, it signifies that the RNA quality in the sample is considered unsatisfactory or unacceptable for use in molecular assays. So here, how can we find the concentration of the nucleic acid, whether it's an RNA or DNA? So here, it's easy. We'll just multiply the the absorbance reading by the conversion factor and the dilution factor. So for example, a DNA preparation diluted 1:100 yields an absorbance reading of 0.200 260 nm. So how can we obtain the concentration in Ug per ml? So multiply the absorbance reading, where is that? This one, absorbance reading by the conversion factor. Why 50? Because remember 50 for DNA. 50 for DNA. See? Absorption constants, 50 for DNA, 40 for RNA. And then times or multiply by 100. Why? Because that's the dilution. Diluted 1:100, so 100 times. So the answer is 1000 Ug per ml. So the yield of the sample is calculated using the volume of the preparation. So if in the case illustrated above, the DNA was eluted or suspended in a volume of 0.5 ml, the yield would be, so your formula will be the DNA concentration in Ug per ml multiplied by the volume. So the answer is 500 Ug. See, it's just easy. Next, for an RNA example, it's just the same, but we will just change the conversion factor. So again, 50 for DNA, 40 for RNA. So multiply the absorbance reading. What's the absorbance? Here, absorbance reading of 0.500 multiplied by the conversion factor and then multiplied by the dilution. That's 200. And then out from this, concentration of the RNA, multiply it by the volume if it was resuspended in a volume of 0.2 ml. So your answer will be 40 Ug in every 0.2 ml of RNA, resuspended RNA. So here, measurement of nucleic acids quality and quantity. So Hirsch dice binding specificity for 80 residues, however, complicates measurements of DNA that have unusually high or low GC content. So a standard measurement is needed to determine concentration by fluorometry, and the standard must have GC content similar to that of the samples being measured. So here comes the calf thymus DNA, and usually is the standards for specimens with a known DNA GC content. And Hirsch dice is very sensitive, up to 200 ng nanograms DNA per ml. But if we're using Hirsch dice for fluorometric analysis, so we need to do calibration. So the dye has to be diluted to a working concentration of 1 Ug per ml in water, and then we need to mix this sample. And after mixing, we should do the reading within 2 hours, because that is the time that the dye sample complex is stable enough to be tested. So other fluorescent dyes that can be used, we also have pico green and oligo green. And pico green is more sensitive than Hirsch, so it's sensitive up to 25 picograms per ml. And like Hirsch, ssDNA and RNA do not bind to pico green. We also have oli green, so oligo green. So it is designed for short pieces of single-stranded DNAs. And the limit of detection is 100 picograms per ml. So going back to cyber green is remember, for RNA. And this one, the intensity fluorescence is less with polyadenylated RNA than with total RNA. So the sensitivity is down to 2 ng per ml. So have you noticed that fluorometric analysis is super sensitive as compared to spectrophotometric and fluorescent? Don't confuse fluorescent spectroscopy from spectrophotometry, okay? And that of the other one. So sorry guys, I'm not really with electrophoresis. I'm having a nosebleed episode, so it's not easy. Sorry. And that's the end of nucleic acid extraction. So please study because a poster shall be given this coming January 13, Saturday, and the time will be announced later. Your mayors will inform you guys. Thank you and have a nice day.